Now let us discuss what is meant by chemical shift, and before that let us find out some important characteristics of NMR spectrum. In NMR spectrum, the frequencies are represented by peaks, and there can be different number of peaks. The number of peaks depends upon the number of protons that is causing resonance. Also in NMR there are different peak positions. The position of the peak depends upon the nature of the proton and the identity of the surrounding atom, to which atom the protons are surrounded. For more clarification, consider the example of methyl alcohol CH3OH. If we observe the NMR spectrum of methyl alcohol, we can see two distinct peaks, one corresponding to OH and one corresponding to CH3. This is taken under a low resolution with the peak intensity 1:3. Why two peaks are coming in CH3OH? That is the question. If all proton absorb energy in the same magnetic field, we will see single peak. Now in nuclear magnetic resonance, we are describing about the interaction of nucleus with the radio frequency region that is placed in a magnetic field. It is actually the protons in the nucleus that absorb the radiation and undergo transition. So if all the protons absorb energy at the same magnetic field, then we will see single peak. However, we can see two distinct peaks in methyl alcohol under low resolution. Then one proton corresponding to OH and one proton corresponding to CH3. We can say it is a CH3 proton. The same thing happens in the case of acetaldehyde also. Acetaldehyde is CH3CHO. In the case of acetaldehyde also, we can see two peaks. One is CHO proton, one peak corresponding to CHO proton and one peak corresponding to CH3 proton. The spectrum is taken under low resolution. From these two examples, we can say that the nature of the pattern, the nature of the NMR spectrum depends upon the environment to which the protons are associated. Since the environment affects the nuclear magnetic resonance pattern, we can introduce a term called chemical shift. Chemical shift is defined as the separation in the position of the spectral lines associated with the hydrogen atom in different environment. The nature of the spectrum will be different under different environment. So we are introducing a shift, a term called chemical shift, that is the separation in the position of the spectral line associated with the hydrogen atom in different environment. That is how much the spectrum of our sample deviate from the standard sample. That measure that gives the chemical shift. So we can define delta as the B reference minus B sample by B reference. So what is B reference? It is a magnetic field at which resonance occurs for the reference sample. B sample is the magnetic field at which resonance occurs for the given sample. So we need a reference sample, and usually a standard material is used as the reference sample. It is a tetramethylsilane CH3,4Si. It will be named as TMS, or it is named as TMS. In terms of frequency, we can define the chemical shift as frequency of the sample minus frequency of the reference by operating frequency. If you multiply this quantity by 10 raised to 6, then your result will be in parts per million. And please remember that these two spectra are taken under low resolution. The condition of the spectrum at a higher resolution will be discussed in the next video.